There is a mystery that lasted for 670 years. Where did the Black Death begin? Not in the general sense, which was never in serious dispute. The plague that killed between 30 and 60% of Europe's population between 1347 and 1351, the single most destructive epidemic in the history of Western civilization, was caused by the bacterium Yersinia pestis, carried by fleas on rats. And it arrived in Europe through the ports of Sicily and southern Italy from somewhere in the east. That much was known. The specific question, the precise origin point, the specific population of rodents or humans that harbored the strain that would go on to kill 25 million Europeans,
this shaped modern
remained
European
one of the most
immunity.
contested unsolved problems in the history of epidemiology. Until 2022, when a team of researchers published a study in the journal Nature that extracted ancient DNA from a 14th-century cemetery in Kara-Djigach, in what is today Kyrgyzstan, and from a neighboring site at Burana. The cemeteries had been excavated in the 19th century by Russian archaeologists who noted that an unusual number of the headstones were inscribed with dates from 1338 and 1339, and that many referred to an unnamed pestilence as the cause of death. The inscriptions had been studied by historians for over a century, without reaching a definitive conclusion about whether the deaths they marked were connected to the Black Death epidemic that would devastate Europe 8 years later. Then the ancient DNA team extracted genetic material from the teeth of 7 individuals buried at the site. Read the genetic code of 700-year-old Yersinia pestis bacteria in those samples, and traced the strain to a specific population in the mountains of Central Asia, with a precision that no previous historical or archaeological analysis had achieved. The strain in the Kyrgyz graves was a direct ancestor of the strains that caused the Black Death in Europe. The timing was right. The location, on the trade routes that connected Central Asia to the Mediterranean world, was right. The mystery of the Black Death's origin was solved, and then the larger questions began. To understand why this matters beyond the specific origin question, you have to understand what the Black Death actually did to Europe. Not just in the years of the epidemic itself, but in the genetic and demographic legacy it left in the populations that survived. The death toll is the starting point, but not the end of the story. Modern historical epidemiologists working from parish records, tax rolls, and other documentary evidence have concluded that Europe's population, estimated at somewhere between 75 and 80 million people in 1347, had fallen to somewhere between 50 and 60 million by 1353. In some regions the losses were far higher. Some estimates for England suggest a population decline of 40 to 50%. Some Italian cities lost more than half their inhabitants. The plague of Justinian, the previous major plague pandemic of the 6th and 7th centuries, had been devastating. The Black Death was something else entirely. It was the closest thing to a continental population collapse that Europe experienced in the historical period, and the DNA of modern Europeans carries the record of that collapse in ways that researchers are only beginning to fully characterize. When population geneticists analyze the genetic diversity of European populations and compare modern samples to ancient DNA from pre-plague and post-plague periods, they find measurable differences that reflect the severity of the demographic bottleneck. Genetic diversity in some European populations shows patterns consistent with the rapid loss of a large fraction of the population and the subsequent expansion of the survivors. A bottleneck event that compressed the genetic variation of the population in the same way that the Irish famine compressed the genetic variation of the Irish population, but on a continental scale and at a depth that the documentary record alone could not have captured. But the most specific and most scientifically startling finding about the Black Death's genetic legacy concerns not the general pattern of population reduction, but a specific and narrow question. Did the Black Death drive natural selection in the surviving European population? Did the specific immune characteristics of the people who survived the plague differ from those of the people who died in ways that left a measurable genetic signature in the descendants of the survivors? A landmark study published in 2022 in the journal Nature examined this question directly, analyzing ancient DNA from individuals who died before, during, and after the Black Death from burial sites in Denmark, England, and other parts of Northern Europe. The researchers were looking specifically at a set of immune-related genes, particularly variants of a gene called ERAP2 that plays a role in the immune system's ability to recognize and respond to bacterial pathogens. What they found was striking. Specific variants of ERAP2 that appeared to confer some degree of protection against Yersinia pestis showed significantly elevated frequencies in post-plague samples compared to pre-plague samples. In other words, the people who survived the Black Death were, on average, more likely to carry the protective ERAP2 variants than the people who died. And this difference in frequency, driven by the selection pressure of one of the most lethal epidemics in human history, is still detectable in the genomes of modern Europeans today. The Black Death, this analysis suggests, was not simply a demographic catastrophe. It was one of the most powerful selective events in the recent evolution of European populations. The plague killed so many people so quickly that the immune characteristics of the survivors were meaningfully different from the immune characteristics of the general pre-plague population. The survivors were not simply the lucky ones who happened to escape exposure. They were, in measurable biological terms, the ones whose immune systems were better equipped to fight the pathogen. And the genetic signatures of that better immune response are still present, at elevated frequencies, in the descendants of the survivors. This finding has implications that extend well beyond the historical question of what the Black Death was and where it came from. It is a demonstration of natural selection operating in a human population in historical time, at a speed and with a specificity that is rare enough to be clearly visible in the genetic data. Most evolutionary changes in human populations happen too slowly to be detectable across a span of only a few generations. The Black Death happened fast enough, and killed selectively enough, to produce a measurable genetic shift in the surviving population that is still present 7 centuries later. There is a dimension of the ERAP2 finding that the researchers themselves noted with cautious specificity. The same variants that appear to have provided protection against Yersinia pestis are associated, in modern populations, with elevated risk for certain autoimmune diseases, Crohn's disease, lupus, rheumatoid arthritis. The immune characteristics that made 14th-century Europeans more likely to survive the plague may have increased the susceptibility of their 21st-century descendants to a different category of immune dysfunction. One in which the immune system attacks the body's own tissues rather than failing to respond to a pathogen. This is a version of a pattern that appears repeatedly in the study of human immune evolution. A genetic variant that is protective in one infectious disease context can be harmful in a different context, or in an environment where the original pathogen is no longer present. The CCR5 delta 32 mutation, which provides near-complete resistance to HIV infection, also appears to confer some resistance to plague. It is found at elevated frequencies in European populations, possibly as the result of selection during plague epidemics. The pattern repeats across the immune genetics of multiple European populations, where the frequency of specific immune variants appears to reflect the history of epidemic diseases, rather than being randomly distributed as neutral genetic drift would produce. The immune characteristics of modern Europeans are, in part, the legacy of the pathogens that killed their ancestor, and the specific autoimmune vulnerabilities that are more common in European populations than in others may reflect, in part, the selection pressures of epidemic diseases that have not circulated in Europe for centuries but left their mark in the immune genetics of the survivors. The Kyrgyzstan study offered one additional finding that the researchers noted as particularly significant for understanding not just the Black Death, but the broader history of plague outbreaks in human populations. The strain recovered from the 1338-1339 cemetery at Kara-Djigach was positioned in the Yersinia pestis phylogenetic tree, close to the root of the diversification that produced all four of the major plague strains that have caused human pandemic disease. This means that the ancestor of all major human plague strains was circulating in Central Asian rodent populations in the early 14th century, and that the Black Death was not an isolated emergence, but the first of several jumps from this Central Asian reservoir into human populations that would occur across the following centuries, including the third plague pandemic of the late 19th and early 20th century that spread from China to port cities around the world, including San Francisco, where it established the enzootic reservoir in American ground squirrels that still exists in the American Southwest today. The 2022 Kyrgyzstan study, which identified the specific Central Asian origin of the Black Death strain, did more than solve a historical mystery. It demonstrated that the bacterium responsible for the 14th-century pandemic was not simply a more virulent version of a pathogen that had always circulated in Europe. It was a strain that had been evolving in Central Asian rodent populations, in the marmots and other ground squirrels of the mountain plateaus, for a period that the ancient DNA analysis suggests was relatively short before it made the jump into the human populations that would carry it westward to Europe. The specific genetic characteristics that made this strain so lethal to European populations may reflect, in part, the fact that those populations had no prior exposure and therefore no prior selection for the immune variants that would have provided some degree of protection. The Central Asian populations who had lived in proximity to the plague's reservoir rodents for generations may have had some degree of pre-existing immune adaptation that European populations entirely lacked. The Black Death was never simply what we thought it was. It was never simply a catastrophe imposed on a passive population by an indifferent pathogen. It was a selective event, operating with the blind efficiency of natural selection on a population that happened to be variably equipped to survive it, producing in the survivors a genetic legacy that is still present in the immune systems of their descendants, shaping the biological vulnerabilities and resistances of modern Europeans in ways that only the most precise genomic tools are now able to read. The plague is over. The pathogen is still present in rodent populations in Central Asia, in the American Southwest, and in other parts of the world, kept in check by modern surveillance and antibiotics that would have seemed miraculous to a 14th-century physician. But the genetic record it left in the human populations it swept through is permanent. It is in the genomes of the living descendants of the survivors, in the specific immune variants that protected their ancestors, in the autoimmune susceptibilities that are the price of that protection. The data is telling us that history does not end when the epidemic ends. It continues in the cells of the people who survived, passing forward through generations, shaping the biology of populations who have long since forgotten what their ancestors survived.